Dice, glaucoma's hidden culprit. Your eye is slowly starving itself, and you don't even know it. Not from lack of food, but from lack of blood. Right now, thousands of people are losing their vision to glaucoma, a disease we've spent decades treating like a pressure problem. We've been lowering the pressure, measuring success, patting ourselves on the back. But there's a darker truth hiding underneath those numbers. What if the real killer isn't the pressure at all? What if it's something far more sinister happening at the cellular level, a cascade of death and inflammation that pressure-lowering drugs just can't touch? And what if, bizarrely, the answer involves compounds that people have been taking recreationally for decades? We're talking about psychedelics. In a new study, researchers are suggesting that psychedelics might be the key to saving your eyesight. So what exactly is happening inside a glaucomatous eye? When we think about glaucoma, we think about pressure. High intraocular pressure, the buildup of fluid inside your eyeball, has been the villain in the story for over 100 years. But medicine is learning something uncomfortable. We've been staring at the wrong monster. Glaucoma is increasingly understood as a vascular disease. It's about blood flow. It's about your eye tissue not getting enough oxygen. And when tissues don't get enough oxygen, something called ischemia-reperfusion injury kicks in, a brutal cycle where cells die, inflammation spreads, and the damage cascades outward like a stone thrown into still water. The retinal ganglion cells, the ones responsible for sending visual information to your brain, are particularly vulnerable. They're like the night shift workers of your eye, hard-working, essential, and apparently first to suffer when things go wrong. Current treatments, which mostly focus on lowering eye pressure, have helped millions of people. But they're incomplete. They're treating a symptom, not the underlying disease, which is why researchers in Croatia decided to look at a completely different toolkit, one involving neuroplasticity, one involving the brain's ability to rewire itself, and compounds that make it rewire very, very quickly. This work comes from a team at the University of Rijeka in Croatia, specifically from the Department of Basic and Clinical Pharmacology and Toxicology, alongside ophthalmologists at the Clinical Hospital Center Rijeka. The lead researchers include P. Dolenec, G. Pelčić, and K. Pilipović, among others. It was published in February 2026 in the journal Pharmaceuticals, which is the peer-reviewed open-access journal where this kind of translational work, bridging basic science and clinical application, tends to land. This is recent work. This is cutting-edge thinking. What's interesting about this team is the collaboration structure itself. They've got pharmacologists sitting alongside eye doctors. That interdisciplinary approach matters because it meant someone could ask the question, hey, what if we stopped thinking about this as just an eye problem and started thinking about it as a neurological problem? That's the move that opens doors. So what's the actual mechanism they're exploring? Here's the key. Glaucoma causes ischemia, a lack of blood oxygen, to the retinal ganglion cells. When blood flow is restricted and then restored, cells don't just bounce back. Instead, they experience what researchers call excitotoxicity. Imagine a neuron as a tiny factory. When oxygen runs out, the factory's emergency systems kick in. But those emergency systems are designed for short-term survival, not long-term thriving. They flood the cell with glutamate, an excitatory neurotransmitter, which, at high levels, becomes a poison. The cell starts firing uncontrollably, consuming its own resources, essentially running itself into the ground. Simultaneously, immune cells in the brain called microglia activate. They're supposed to clean up damage, but in glaucoma they become overactive and start attacking healthy tissue too. It's like calling in a cleaning crew that shows up and bulldozes the whole building instead of sweeping the floor. Meanwhile, astrocytes, star-shaped cells that support neurons, also activate in a harmful way. The result is chronic neuroinflammation. The eye tissue is in a state of perpetual low-grade war with itself. Standard pressure-lowering drugs don't address any of this. They don't heal the cells, they don't calm the immune response, they don't rebuild connections. Which brings us to the strange intervention the researchers are studying: psychoplastogens. Psychoplastogens are a class of compounds that do something almost miraculous. They rapidly enhance structural and functional neuroplasticity, meaning they help the brain and nervous system rebuild themselves, grow new connections, and repair damage. The compounds studied include ketamine, psilocybin, the active ingredient in magic mushrooms, and DMT, the compound found in certain plants and some people's cerebrospinal fluid. There are also newly synthesized non-hallucinogenic analogues, which is important because it suggests we might get the therapeutic benefits without the perceptual side effects. Here's the part that blew my mind. These compounds work through specific biological pathways. The BDNF-TrkB-mTOR pathway. BDNF is brain-derived neurotrophic factor, essentially fertilizer for neurons. When this pathway activates, neurons grow dendrites, the branching structures that allow them to connect with other neurons. Synapses form. Neural networks become denser and more functional. The retinal ganglion cells, instead of withering under the stress of ischemia, start rebuilding. They start reconnecting with the visual pathways in the brain. But that's not all. Psychoplastogens also modulate glial cells. They calm down the overactive microglia. They rebalance astrocytes. They dial down the neuroinflammatory response. In essence, they attack the glaucoma problem from multiple angles simultaneously, supporting neuronal survival, promoting regeneration, and suppressing the immune attack that's perpetuating the damage. What makes this findings genuinely significant is the convergence. It's not like psychoplastogens help with one aspect of the disease. The research suggests they address multiple mechanisms simultaneously. The excitotoxicity problem gets better because neurons are more resilient and connections are forming faster than they're dying. The neuroinflammation problem gets better because glial activation is dampened. The lack of neuronal connectivity gets better because the BDNF-TrkB-mTOR pathway is driving synaptic growth. It's a multi-target intervention in a disease that clearly needs multi-target intervention. Now, a crucial honesty check. This is a review paper, not a clinical trial. The researchers are synthesizing existing evidence and making the case for why this avenue is worth exploring. They're not saying psychedelics cure glaucoma. They're saying the mechanism is there. The biological logic is sound. And critically, compounds with these properties, compounds that enhance neuroplasticity and modulate neuroinflammation, have a much better shot at actually slowing or stopping glaucomatous neurodegeneration than pressure lowering alone. Effect sizes and clinical efficacy remain to be determined. But the direction of thinking is important. What does this actually mean for someone with glaucoma right now? Probably nothing, honestly. We're not at the clinical trial stage for psychoplastogen-based glaucoma therapies. You shouldn't stop taking your eye drops or seeing your ophthalmologist. What this means is that the field is expanding its conception of what glaucoma is, and therefore what might treat it. Instead of viewing glaucoma as purely a vascular, mechanical problem, researchers are viewing it as a neurodegenerative problem. And neurodegenerative diseases, it turns out, respond to interventions that restore neural plasticity. The honest limitations. Psychoplastogens have their own risks and side effects. Hallucinogenic compounds raise obvious concerns about adverse effects and abuse potential, though the non-hallucinogenic analogues sidestep this. The blood-brain barrier complicates drug delivery to the eye. And we don't yet know optimal dosing, timing, or whether benefits would persist. But the fundamental insight is solid. Neuroinflammation and loss of neuroplasticity are real features of glaucoma. And compounds that address those features are scientifically reasonable candidates for further investigation. This sits at an interesting intersection in neuroscience right now. There's a broader movement recognizing that neuroinflammation and reduced neuroplasticity are underlying problems in many neurodegenerative diseases: Alzheimer's, Parkinson's, even depression and anxiety. Compounds that enhance neuroplasticity, particularly psychedelics and their analogues, have moved from fringe to mainstream in research over the past 5 years. Clinical trials for psilocybin in treatment-resistant depression are actively recruiting. Ketamine is already FDA approved for treatment-resistant depression in nasal.